Okay. Okay, and another small practical detail. So if you would need to have a certificate of attendance, please let us know by providing your name and email address also in the chat. Because for Brussels environment, if you follow all 4 webinars of Replace, this will count as training. So don't hesitate to put the details in the chat, and then we will be able to provide you with this certificate. So to start, I would like to introduce you with the first speaker, which is Professor Cleo Goyvaerts, who works at the Vrije Universiteit Brussel. So she will present her work on the development of a 3D lung tumor spheroid platform. So Cleo obtained her bachelor degree in Veterinary Sciences and a master in Biochemistry and Biotechnology at the University of Ghent. Cleo then started her PhD at the VUB on antitumor vaccination. She then worked as a postdoc where she focused on the manipulation of the tumor microenvironment in collaboration with the Mount Sinai School of Medicine in New York. And since 2018, she is an assistant professor at the VUB, where she focuses on resistance mechanisms that hamper curative immunotherapy for lung cancer. Cleo, the floor is yours.
Okay, thank you very much, Mieke. All right, I will start by sharing my screen. Okay. Do you all see my screen as it is right now, shared?
Yes.
Yes. Okay. All right, let's kick off then. So thank you for the kind introduction, Mieke. So I will not elaborate further on that. So what I want to talk to you guys about today is about our development of a 3-dimensional lung tumor model that we use now on a daily basis for immune oncology assays. And I'm going to try to talk you through the why we have done this and what the application possibilities are. So let's start off with telling you a little bit more about tumors. I think I can speak for most scientists that up to 20 years ago, most research focused on the tumor cells, which was also quite obvious. If you look into, for example, testing chemotherapy, you just want to look at how are the tumor cells killed. Same for radiotherapy. If you look at surgery, you want to take out the tumor. So it was all about the tumor cells. But more and more research revealed that a tumor is not just a bunch of tumor cells, but it's actually a very complex tumor microenvironment, as it is termed, that is packed with much more than malignant tumor cells. It is actually also packed with non-malignant cells, healthy cells, like stromal cells, fibroblasts that produce extracellular matrix, which provides a framework for the tumor cells to grow and to attach to, but also a lot of different types of immune cells that could help the patient actually conquer its cancer, but on the other hand, also cancer promoting immune cells, which are of course an interesting target for immunotherapy. And especially for the solid cancer, it became pretty obvious that after a while, these solid tumors become almost organs because they also attract blood vessels, lymph vessels. There are even lymph nodes being or sometimes introduced into the tumor. So it's a very complex tissue that also deserves then a more complex approach to study. The issue then, of course, is if we think of the tumor microenvironment and looking into the immune cells that interact with the tumor cells, the issue was that then actually one of the only ways to look into this was by using mouse models, because a mouse model is able to, upon growth of a tumor, also indeed result in the infiltration of immune cells, because don't forget, the immune system is a system, not an organ, that is comprised of the bone marrow, that is the source of all the immune cells. But then every organ has its own specific immune cells. Your spleen is packed with them, your lymph nodes are packed with them, your blood is full of immune cells. So you can imagine that it's not so obvious for an immunologist to look into the immune system interacting with the tumor in vitro. So we were pretty dependent on mouse models. Now, what we tried to do was take out the different cells of a tumor and, for example, sort them in their different cell subsets, being, for example, a tumor cell, but then also looking into the macrophages, the myeloid cells, such as the other myeloid cells, neutrophils or dendritic cells, T cells, lymphocytes, whatever. So different types of cells that can enter the tumor microenvironment, and try at least, for example, separately in a petri dish upon, for example, also their immortalization, to look what is going on. And then, of course, try to sometimes reconstruct their interactions by at least co-culturing them. Now, you have to realize that especially immune cells like macrophages, myeloid cells, they really like to adhere to plastics. So instead of often interacting with tumor, they started to interact with the plastic. They're also immune cells, so they're very sensitive to their environment, and they started to change their character. So it was not always that relevant to study immune tumor interactions in vitro, especially not when they were 2D cultured. And as it is also with other instruments, thinking, for example, of a television, you can have all the different components of a television, but that will not give you a movie. It's by assembling them in the right way that you get your movie and that you get an idea what is going on in your movie. Well, let's say it's actually exactly the same with a growing tumor. You can have all your different components, but it's only when you put them together in the right ratio and in the right co-culture environment that you can really start appreciating, for example, what chemotherapy also really does. Because we have been seeing that although chemotherapy was approved in vitro to kill tumor cells, that in the end, often patients relapsed, amongst other, because immune cells can actually also aid in the resistance that is installed by the tumor cells to chemotherapy. So the bigger or more complete your picture is, the more relevant research we can do into breakthrough cancer therapies. And to give you an idea, in the past 10 years, quite a lot of progress has been made on the use and the design of three-dimensional in vitro tumor models. If you look at that, when in 2006, there was almost no publications, and today we have quite a number of publications. It's a vastly steep increase. Very nice. I don't say that it's completely linked to that. There are, of course, different reasons why the use of laboratory animals went down, also in Belgium, as these are the numbers from VUB specifically. But we clearly also see that there's a decrease in the use of laboratory animals. And I can speak for our lab, who has always been focusing on anti-tumor immunotherapy, using a lot of laboratory animals to sooner or later validate our in vitro findings. And I can assure you that indeed right now we more often start with three-dimensional models. And if we don't see what we want to see, let's say, and we don't see the impact of the therapy, we don't even go to in vivo anymore, while before that was our only way to test our therapy. Now let's talk a bit more about the specific model that we developed. Perhaps a bit of background information. So what is a very typical way today to grow organoids that's based on patient material or spheroids is by basically just culturing one or a few cells, the same subset, in an ultra-low adherent plate or in hydrogel. And by themselves, they form then units. Now, what our problem was that it's still a monoculture. It's just a 3D grown monoculture. And that's not what we wanted. We wanted a system where there was a stromal component that was relevant for the in vivo situation. So that's why instead of using a hydrogel, which is actually a mix of extracellular matrix, we opted to test the addition of fibroblasts, which are also often present, especially in lung cancer, to then co-culture them with tumor cells, which we stained in this case with an enhanced green fluorescent protein to follow them up over time. And the rationale was that these fibroblasts would start to produce extracellular matrix themselves, and as such actually provide this architectural framework for the tumor cells to grow in. To give you an idea, this is a non-small cell lung cancer patient biopsy, where I'm sorry for not putting the legends here, but in green we have keratin staining for the tumor islets that you can see, all those green islets. And in between you see in red alpha smooth muscle actin, which is a typical marker for extracellular matrix. And you nicely see this islet stroma division. Important, it's, for example, this stroma that makes it difficult for immunotherapy to do his thing, because often immune cells just get stuck in this stroma, which is something, if you have a monoculture, you will never appreciate. Now, when we used our technique, and I think the most important thing here to mention is that the optimization of this model lies in defining the exact ratio of fibroblasts and tumor cells you need to use. And as a side note, this is different for different tumor cells. And we found for 3 different lung tumor cell lines, so 2 murine cell lines and 1 human cell line, that if we mix them at a specific ratio, especially for the KP spheroid model, we nicely found back again these tumor islets. In this case, it's not the keratin staining, but it's the green fluorescent tumor cells that you find here back in the islets, and that you get then these borders of stroma that have been produced by the fibroblasts. For the Lewis lung carcinoma model, who is known to have more of an epithelial to mesenchymal cell transition profile, we saw a much less obvious division of green versus red. It seemed like they almost acted as fibroblasts themselves. While for the H1650 model, again, we saw that there were more organization of green islets dispersed with then red zones of stroma. So this gave us a first, in our opinion, more relevant way to start studying now also what now the impact is of adding immune cells to these lung tumor model. So first thing we asked ourselves is, well, what happens now with macrophages if we add them to these cultures? So we know that macrophages, if we would add them, we could add 2 different types, because in a way we wanted here to make it as extreme as possible to kind of see what happens. If we add, we call them the good and the bad macrophages. Of course, this is a vast oversimplification, but it's to give an idea. So the M1 macrophages, if you find these back in the tumor, they're often seen as the good ones, the ones that stimulate an immune response and that aid in the killing of tumor cells. While the M2 macrophages are more the immunosuppressive macrophages that are often linked to resistance to immunotherapy. Now, what we did is in vitro, we generated our spheroids, and we generated these 2 different types of macrophages, the good ones, the bad ones, and then looked into how did each of them interact with the tumor microenvironment. Was it similar or was it different? And to our surprise, we found that it was very different. While the M1 macrophages, most of them didn't even survive in the culture over time, and the ones that were still alive were mainly found on the outside of the tumor. It looked like they were almost incapable of entering and infiltrating the lung tumor spheroids. While for the M2 macrophages, it seemed like they were much better and capable at really interacting with the tumor, and probably also because they are more related to tumor progression, aid in the further proliferation of the tumor cells. So these images were made upon fixation of the spheroids and slicing them, because of course that's the nice thing of a 3-dimensional model and no longer a 2D planar tapestry. You have something with which you can work also afterwards. It's 3D, you can fix it, you can stain it, you can slice it. A next application was looking into T cells. So T cells and how they are involved in the tumor microenvironment. And again, to give you an idea, this is a non-small cell lung cancer patient biopsy. And in this case, you can see that in green, again, in keratin, the keratin stained tumor islets. In this case, in gray, you have the stroma, so the alpha smooth muscle actin. And in red, all these small dots are T cells. And as you can appreciate, you can see that the T cells really get stuck in this stroma, and in this case, hampering them from basically recognizing and killing the green zones. Now, this is the spheroid model that we then obtained after adding also these T cells to our model. And as you can see, it was pretty similar. Again, the T cells getting mainly stuck in the stroma and almost not able to interact with the green islets. Now, of course, what happens if we then combined both of them? Especially, of course, looking into what do the M2 macrophages aid in the tumor progression? Do they interact with the T cells? And the nice thing again is, if we have a 3D culture, it's a small culture, you can look into the spatio-temporal analysis. Again, something, and I think that's an important note. I don't want to say that everything needs to be replaced in the future, because it will always be difficult to replace every in vivo situation in vitro. But it's worth mentioning that some in vitro tools actually give you more information than an in vivo tool. For example, in this case, if you look here into the, try to see if I can perhaps do it like this. No. Ah, yeah, okay, sorry. Yeah, there it is. So we can also make movies, which is much less obvious in vivo to do. This is a loop. I think it was a movie made in 20 minutes. And as you can see, for example, here in gray, these are T cells. In this case, sorry, sometimes the colors mix. But in this case, the small gray cells are the T cells. And you can see, for example, here they try to fight off a green cancer cell. Unfortunately, all the other T cells, or most of them, are in more close proximity with the M2 macrophages instead of with the tumor. So again, showing that in this case, it will be much more difficult for the T cells to focus on the cancer cells, because they probably get manipulated by the macrophages. Okay, like this. All right. So then the final application I want to share with you today is the fact that we could also use these 3D-dimensional models to study tumor cell-specific killing by cytotoxic T lymphocytes. In a way, the most important readout if we also want to evaluate new immunotherapeutics, which are often based on stimulating cytotoxic T lymphocytes activity on tumor cells. So for that, we took another approach. So again, of course, we mixed fibroblasts with tumor cells. But what did we do this time? We mixed tumor cells with a green color, but also that were seen as target cells for the T cells, next to non-target cells. So in this case, these were also cancer cells, but they were not recognizable. No? We mixed them in our cell repellent plates. Around 8 hours later, we added the cytotoxic T lymphocytes that could specifically kill the green cells. And as you can see over here, this is then the picture you get. Here, the fibroblasts are not stained. The nice thing is, again, then, you can basically fill an entire 96 well plates with the spheroid, so you really already have nicely your replicates. You don't need 96 mice. You can immediately go to 96 wells without bothering any animal. And the nice thing is that after adding these T cells, a few days later, you can measure what the specific ratio is of your green and your red cells. Yeah, so here you have the red signal, you have the green signal. And after 5 days without T cells, you just have around 20%, in both cases, 70–80% of your red and your green cells, next to your fibroblasts, of course, in your spheroid. But when the T cells were added, you have a decrease of your EGFP positive cells specifically. Yeah? And by combining this ratio with this ratio, you can very specifically measure the target cell specific killing. Something that is nice because it gives you a very sensitive readout, which you really need for deciphering if your immunotherapy gives a better result than, for example, the standard of care. And you can, of course, also use a supernatant to look into cytokines that are secreted by the T cells. And again, last but not least, if you look into the spatiotemporal follow-up opportunities. So here, it's not like a high-resolution image as for the confocal microscopy images I showed earlier. Therefore, we use incucyte live cell imaging to see what happens over time if T cells are added to spheroids and are capable or not capable of killing green cells. So I'm just going to show you here what is going on in a spheroid without T cell addition. And you see over time that, especially in the first days, these spheroids actually decrease in size, which is typically or has been described previously to the fact that you have more tight junctions and extracellular matrix that in first instance concentrate your spheroid more. Now, here we have spheroids to which we added cytotoxic T lymphocytes, but in suboptimal conditions, so without a specific activation of the T cells, so without treatment. And as you can see, this is when the T cells are added to the culture. And I want to ask you to look at the image, but also at the timetable here below, which shows you that, yeah, we're following up these spheroids now over a number of days. At day 3, we're here now, and you can appreciate that here most tumor cells, the green cells, the green target cells, died. However, after day 5, they got back, which is for us, of course, a beautiful model to now start looking into resistance mechanisms, because it seems that even though you have good cytotoxic T lymphocytes, if they are there in suboptimal conditions, you also get the initiation of resistance. Now, if we did the same, but then with optimally treated and stimulated T cells, this is what we got. So you can again see T cells are added, and we're not even 1 day further, and all green cells were dead. And the good thing was, in this case, they didn't get back. So I think this essay shows that there is definitely power in 3-dimensional models to study T cell interactions, macrophages, and immunotherapy. So with that, I like to conclude my talk. I think as take-home messages that we can say that this is quite a straightforward way. It's not difficult to basically mix the cells once you have them, of course, at hand. It can fairly recapitulate the characteristic tumor islet stroma architecture. We tested it for macrophage and T cell infiltration studies, but of course, this expands much more application possibilities for other groups. It's suitable for spatial temporal tumor cell specific killing evaluation, something, again, I want to stress is impossible in vivo. It can go in a 96 well format, so you have a lot of different wells at your disposal. And we, of course, used in non-small cell lung cancer, but we do believe that this can also serve as a blueprint for other solid cancers. We already tested this for melanoma, giving us also nice results. And of course, this technique was added to the replace database. So please feel free to go and visit this database if you want to have more information about the model. And last but not least, of course, I want to thank all the collaborators who worked on this this model, especially special thanks also to Professor Hélène Salmon from Institut Curie, who together with me actually started this research at Mount Sinai in New York now about 6 years ago. So voila, with that I want to conclude, and of course I'm open to any questions.
So thank you very much, Cleo, for this nice presentation. I think it's really a good example on how we are evolving in immuno-oncology research. So first we can see if there are any questions from the audience. I can unmute you if you have a particular question.
I see a question from Julie Sanders.
Yes.
On how do you see the future for this model of evolving? I think the future depends on the questions, the research questions of the researcher. So in our case, for me, it's always important to do relevant research. So on the one hand, we have our cell lines, of course, but it would be even more elegant if we could move this further to patient-derived material. Of course, I didn't go into the details here, but we used an immortalizing large T antigen SV40 encoding lentiviral vector to design our fibroblasts, to make them immortal. And it would be great if we could actually try to mimic this process also with patient materials. So we kind of, for example, make cell lines out of the fibroblasts or the cancer-associated fibroblasts that we isolate from the tumor tissue, and then again also co-culture them with their own tumor cells. So it's not just an organoid based on one stem cell-like cancer cell, but it's again starting to be a co-culture. Because I think another thing that I do want to mention here is that we are always very dependent on the histocompatibility of the models if we go into immunotherapeutic research. So in this case, we were in the end confined to the models with a histocompatible nature. So for example, for the human model, everything was HLA-A2. But again, this is not always further extrapolatable for all other cell lines and fibroblasts. So for me, this is definitely something I'm kind of dreaming of, that we can extrapolate this and no longer use hydrogels, but also use patient-specific cuffs to do co-cultures and further reveal the tumor complexity per patient.
Okay, thank you. I can see we have another question from Nicolas Bayens. What about hypoxia and the lack of perfusion?
That's definitely also a very good question. So the thing was that in our hands we had very small spheroids. So again, something you can for your own purpose or your own research question, you can play around with that. But sometimes our spheroids were only about 200 to 300 micrometers in size, and then the hypoxia in the core was relatively small. So because actually that was one of the questions of our reviewers after submitting our paper for publication, and we used pimodidazole to look into hypoxia, but it was rather small. Of course, if you want to go into bigger spheroids by using more cells and also following them up over time longer, this is definitely an issue. We have a collaboration ongoing with microfluidics experts from Neuchâtel, Switzerland, because this is indeed also something important to take into account. So you have these microfluidic models where there is indeed a higher oxygen rate that can be perfused over a period of time. And of course, especially if you think, again, I'm sorry, I'm always into the patient-derived culture because that's something that really interests me. But what is there often considered is these air-liquid interfaces where they are really more in contact with air, because in the end, you really submerge your cells, 2D or 3D, whatever, always in medium. And in a way, you are drowning them, because that's not the situation in vivo either. There they are being oxygenated via vascularization. So this is something we definitely want to go to as well with our 3D models.
Okay, thank you. Are there any other questions from the public? I don't see anything in the chat, but I can also unmute you if you have a particular question for Cleo. I don't see.
There is another question.
Yeah.
For the non-tumor studies,
For a beginner.
Yeah. Of course, the thing is that everything in research in general, it's often an entire organ. There's also other cells involved. If I think, for example, of liver, we have a group here that focuses on liver and on also fibrosis in liver. Well, they are using a similar approach, is maybe a big word, but they also use both fibroblasts and then also the in their case more stellate cells and hepatocytes, of course, to mix with them. In the end, they often work with cell lines, so they are capable already of being grown in vitro and can then be mixed. But it's always a matter of finding the right ratio, the right culture conditions, of course, and that's the tricky part. But the cool thing is that there's so much research ongoing that today you can kind of start already from a protocol that you will find in literature for your specific field of interest. And of course, then, because I think that's always a thing, what is your research question and which model can you use to answer that one the best? Not just we use a 3D model because that's cool or that's one. No, why do you use that? In case of immunotherapy, you just really don't have another option because you need co-culture of these cells to be able to measure immunotherapy impact. You cannot do it in co-monocultures. So anyway, that's I think very important as a researcher always. What is my research question and what is the best model out there today that can help me answer my question?
Okay, thank you, Cleo. Who's 9? I'm not sure if this answers your question fully. I can maybe unmute you. Search.
Of course, there are a lot of in-depth reviews available for different fields.
Thank you very much.
Sorry.
It was good enough for me.
Okay.
Great.
Thank you for your question and thank you for your comments. Are there any other questions from the public? Otherwise, we will continue with the next speaker. I don't see any coming in. All right. If you still have a question, you can still put it in the chat and then we can send you the response after the webinar. So our second speaker of today is Marguerite Meeremans from the University of Ghent, and she will present the in vitro tendinopathy model that she is developing as part of her PhD thesis under the supervision of Professor Catharina De Schauwer at the Veterinary Stem Cell Research Unit in Ghent. So her work focuses on the development of a representative tendon injury model by combining equine cells, gelatin, 3D printing, and mechanical stimulation. And then they hope to better understand the underlying pathways of the pathogenesis and also to see if new therapies can be developed with reducing the numbers of experimental animals. So Marguerite, the floor is yours.
Hello, good afternoon. I'll also try to start with sharing my screen.
It's not working.
Which is taking quite some time at this moment.
Okay.
I hope it will show up.
Click on the icon, yes?
Yes. It's a lot slower.
You don't see anything yet?
No, it's loading. It's trying to make the connection and then it says there's a problem with sharing. I'll just refresh the meeting.
Yeah. Yes. Okay, so waiting a few more minutes. In the meantime, I would like to thank again Professor Cleo Goyvaerts for her presentation on the in vitro model on immune oncology. So thank you very much, Cleo, for your presentation.
You're very welcome and thanks for the invitation.
No problem. Okay, in the meantime, Marguerite is back.
Yes, now it's really working.
Yes. Yes, we can see your screen.
Okay, and still hear me as well?
Yes.
Okay, so good afternoon. Thank you for the introduction. And as you were already introduced to, I will present to you the part of my PhD project, which is the development of a representative in vitro tendon injury model. So first, I would like to start with giving you some numbers. So in Belgium, 3 out of 20 people occasionally go for a run, but 1 out of those 3 runners will develop an Achilles tendon injury. And comparable in horses, 1 out of 5 racehorses will develop a tendon injury during its career. Both the Achilles tendon in human and the superficial flexor tendon in horses are quite similar in function and structure and are frequently injured during exercise because they're submitted to high mechanical strains. These tendon injuries can occur suddenly in healthy tendon, but more frequently there's a chronic degenerative process and therefore the tendons are more vulnerable to little overstrain injuries. So, due to the similarities in function and structure and pathophysiology, the equine superficial flexor tendon is a best animal model at this moment for the human Achilles tendon injuries. Healthy tendon mainly consists of specialized fibroblasts called tenocytes. And these tenocytes are responsible for extracellular matrix production. The tendon extracellular matrix mainly consists of water and collagen type 1. This collagen is hierarchically organized from fibrils into fibers into vesicles. And each vesicle and group of vesicles is surrounded by some loose connective tissue, which contains blood vessels, lymphatics, and nerves. However, healthy tendon is quite hypocellular and hypovascular in comparison to injured tendon. So upon injury, there's an influx of inflammatory cells. There is new blood vessel formation and capillary ingrowth during the inflammation phase. In a subsequent proliferation phase, there is new extracellular matrix being formed. However, this mainly contains collagen type 3 instead of collagen type 1, which is less elastic and more stiff in comparison to the normal collagen. Then in the final remodeling phase, this collagen type 3 should be replaced by collagen type 1, and these collagen fibers should be realigned. However, in adults, this process is never completely completed, and therefore there is scar tissue remaining, and the tendon is more vulnerable to new re-injuries, and we see reduced performance in both human and horses. In both species, the therapy at this moment mainly consists of physical therapies such as schooling or rehabilitation in combination with anti-inflammatory medication and surgery if desired. Although the pain is reduced and this is anti-inflammatory, there is no regeneration of the tendon tissue, and still the scar tissue is still being formed. Therefore, novel therapies such as laser, shockwave, or biological therapies are being developed at this moment. And in our lab, we're focusing on the mesenchymal stem cells, or MSCs. These MSCs are multipotent adult stem cells, which you can obtain from various sources, and they have a great regenerative potential due to their capacities to restore tissue and also to reduce inflammation. In horses, MSCs are already frequently used. However, their widespread clinical application is being hampered by the fact that there are no long-term controlled, placebo-controlled randomized clinical studies in both human and in equines. And therefore, the long-term effects are not are unsatisfying. And there are still some practical questions remaining, such as how much cells should we give and when. So therefore, we're developing an in vitro tendon model to study these MSCs in a representative environment. And now I'm going to walk you through the development of the model. So first, we need to capture the relevant cells. So therefore, equine tendon cells are isolated from the superficial flexor tendon, which you can see on the picture. And simultaneously, we also isolate and purify equine endothelial cells from the digital vein, which is located just medial from the superficial flexor tendon. And as you already heard before, immune cells are very important. So also in our model, we're implementing these immune cells. So we isolate peripheral blood mononuclear cells using density gradient centrifugation, after which we purify these cells and we only keep the monocytes, which we differentiate in 8 days in culture into macrophages. We also have the good and the bad ones. However, in our case, the roles are switched, and the M1 macrophages are the bad ones and the M2 macrophages are the good ones. So you can see them here in culture. They have a quite typical morphology in 2D cultures, and we also characterize them using RT-qPCR. Second element of our model is the extracellular matrix. So as I said before, healthy tendon consists of water and collagen. So therefore, we're using gelatin-based hydrogels to mimic this environment. We're using gelatin methacrylate and gelatin norbornene. Hydrogels contain large amounts of water and are therefore good to mimic the natural ECM. And gelatin is a collagen derivative, which is easy to modify chemically to tune its mechanical properties and to work in vitro. It's also quite cheap and it's widely available. So after chemical modification, we also characterize these hydrogels, of course. For example, using the swelling ratio. The higher this ratio, the more water the hydrogel contains, and therefore the higher the ratio, the better in mimicking the ECM. We also use the storage modulus, which is a measure for the stiffness of the gel. So the higher the storage modulus, the stiffer the gel. And then we also have gel fraction, and it's when it's higher than 90%, it's indicative for a 3D network that is being formed. So after gel characterization, we optimized cell concentration and we encapsulated 5 million tendon and endothelial cells within these 2 gelatin-based hydrogels. And we also included collagen as a control condition. And as you can see on the graph, the viability was above 70% for all conditions, which indicates excellent cytocompatibility. So we continued with the next parameter, which is cell elongation. The equine tendon cells are located between the different fibrils, and therefore they're very elongated into the direction of the tendon itself. And this elongation is crucial for their function. So to quantify this elongation, we use the nuclear shape. So the higher the aspect ratio and the lower the circularity, the more the cells are elongated. So regarding aspect ratio, there were not much differences observed. However, in circularity, we see a much lower circularity in the gelatin norbornene in comparison to the collagen. So the cells were more elongated in the gelatin norbornene. In vivo, you can imagine that the tendons are subjected to mechanical loading every day. And therefore, to make our model representative, we also include this mechanical stimulation. So you can see here our device, it's the TC3 bioreactor from Ebers. And we implemented in these experiments a protocol of 5% strain 30 minutes a day, which is representative for 30 minutes a day trotting of a horse. The cells remained viable when this mechanical stimulation was implemented. So next, we again quantified this elongation. And you can see again, overall, the cells elongated more in the gelatin norbornene. Also, when the cells were added to the gelatins in co-cultures. So first, we investigated the monocultures, and then we included these co-cultures. I'll also show you some pictures. So here you can see the tenocytes elongating in a gelatin norbornene. We also have the endothelial cells and the co-cultures. However, it's important to say that this elongation is more clearly observed in the superficial layers of the hydrogel. And in the center of the hydrogel, they remain quite round. So to prevent this, we looked into the next parameter, which is the nanometric structure. As I said before, the collagen is hierarchically organized, and we try to mimic this organization using electrospinning of a PCl, so it's polycaprolacton. The fibers we obtained had a microdiameter of more or less 10 micrometer, which is representative for the fibril collagen diameter. And on the picture, you can see that in green, the tendon cells elongated into the direction and of the group of fibers, and their angle, they followed each fiber, the angle of each fiber. However, polycaprolacton is a synthetic material and it's not cytocompatible, so we wanted to move to gelatin again. So here, using 3D volumetric printing, we were able in a few seconds to obtain a cylinder. Although the cylinder itself is still a bulk material, inside the cylinder we saw fibers of 5 to 10 micrometers, which is again representative for the collagen fiber diameter. And in the next coming months, I'll also include the tendon cells and the endothelial cells within these cylinders, which will hopefully lead again to clear elongation of the tendon cells. Important next parameter is the damage. So we want to develop an injury model. As I told you in the beginning, the tendon injuries can occur on overloaded, so chronically degenerated tissue, which we will mimic in our model using mechanical overstimulation. And then the tendon is more vulnerable to an acute injury, which we will mimic using a scratch assay. This scratch assay is quite known in literature, and we first optimized this on 2D tendon cell cultures. So here you see a monoculture of tendon cells in a confluent monolayer. And then at time point 0, we made the scratch using a blue tip, and we studied the cell migration over time, as you can see in the pictures. And then we were able to plot the cell migration on a graph, and we can study different conditions using this technique. So to summarize, in the first half of my PhD, I was able to isolate the different cell types. We were also able to include the extracellular matrix composition and try to mimic the structure. We included physiological mechanical loading, and we also were able to mimic the acute damage to the tendon. However, there's still some work to do. In the coming months, I'll try to include mechanical overstimulation. I'll add the cells in the gelatin volumetrically printed 3D cylinders. And we also need to cover the neurovascular supply, which we already covered with the endothelial cells, but we will also include medium perfusion to our model. So after this optimization, we'll use the model to study, for example, tendon pathophysiology, which I will do in the second half of my PhD. We'll use metabolomic and lipidomic analysis to clarify the tendon pathophysiology. And then we'll also try to reveal the therapeutic potential of the mesenchymal stem cells, and more specifically their secretome products. MSCs, as I told you in the beginning, can be isolated from various sources, and their regenerative capacities are strongly cell and context dependent. For example, adipose-derived MSCs have a great anti-inflammatory and immunomodulating capacities, while neonatal sources such as umbilical cord blood or umbilical cord matrix MSCs have more differentiation potential. So to study the best mesenchymal stem cell source for tendon treatment, I already isolated different sources of MSCs. And in a proof of concept, first in some preliminary experiments, we included these different sources in our scratch assay. So we captured the conditioned medium, which is containing all the secretome products of the MSCs in the scratch assay, and we're studying scratch closure. So in the future, in our model, it will become possible to study tendon pathophysiology, and novel therapies such as mesenchymal stem cells and their secretome can be studied in a controlled and representative environment in order to help both human and equine patients. So I would like to thank my two supervisors, all the colleagues in the lab. I would like to thank the FWO for their funding, and of course the organizers for this great opportunity. And then I'm happy to take some questions now or later via email.
Thank you, Marguerite, for your very interesting presentation. It seems that you have already done a lot of work in the first part of your PhD thesis. It was also nice to see that you already took into account acute injuries and chronic injuries, because that was something I did not expect. So maybe if there are any questions, I see.
Yes. There is one question. Are you planning also to test secretome or exosomes?
Yes, in the end, if our model is optimized, we'll first try to include the conditioned media in complete form. And then, of course, the next step will be to look closer into what does the conditioned media contain and what is the most beneficial effect in our model, being the vesicles itself or some specific cytokines, or we want to purify the effects we see with the MSCs and then have a more pure product to hopefully help patients.
Thank you. Does that answer your question, Prashant? If not, don't hesitate to let us know. We can also unmute you if you have... yes, to answer his question. Any other questions from the public? Mo, did you see others in the chat?
I think it's...
Yeah. All right. No. So in the meantime, I would also already like to thank you, Marguerite, for your presentation. So if we receive any questions, we will forward them to you. I would also like to thank the participants for attending this webinar. Our next webinar will be held on the 13th of September. And if you still need to have a certificate of attendance, please let us know by providing us your email address so we can forward it to you. If you are from the Brussels region, this will count as half-a-day training if you participate to all webinars. At the end of the session, you will also have the opportunity to complete a survey about this webinar if you would like to see particular topics addressed, or if you are interested in presenting your work, don't hesitate to let us know. And for the rest, I wish you a very nice and relaxing summer, and we hope to see you again in the autumn when our webinars will continue. Thank you very much to all speakers and of course to you as a participant.